Great. So on behalf of the entire Seven Bridges and the Cancer Genomics Cloud teams, I'd like to thank each one of you for joining us today. As you may be familiar, Seven Bridges is a global biomedical data company which provides end-to-end informatics infrastructure for several disease-focused research initiatives. One such initiative is this cloud resource funded by the National Cancer Institute. It's called the Cancer Genomics Cloud, or CGC. CGC allows cancer researchers across the globe to explore and analyze massive public data sets like the Cancer Genomics Atlas, TCGA, proteomics data from CPDAC and other sources, and several other large multiomics data sets on a secure and scalable computational platform. I would like to acknowledge our funding agency, NCI, and all the Seven Bridges team members who have contributed to the CGC effort, and of course, the users and collaborators who support us. All the resources, such as the slides from each session and the recordings, will be made available at the CGC website, cancergenomicscloud.org. If you go there, you will find the recordings and slides after the session today. If you have any questions, please use the chat box, and after every session, we will answer all your questions. So as you all know, cancer is a complex disease, and it's not possible to understand the different aspects of cancer by studying just one area of the disease, right? So today's session is going to be power-packed with several aspects and data modalities that are required to study cancer. So the first session is going to be on epigenomics. It's titled Epigenomics on the CGC, Enabling Scalable Analysis Using Cloud Computing. And this session is going to be led by expert speakers, Dr. Jeffrey Grover and Nevena Vukovic. Jeff is a genomics scientist at Seven Bridges. He works across teams to expand our bioinformatics analysis offerings, support different programs, and contribute scientific expertise in computational genomics and molecular biology. Nevena is a bioinformatics analyst at Seven Bridges. Today, she is going to be talking about the tools that she led and helped with the entire development during the past several months. She contributes to developing tools and workflows which are made available in the Seven Bridges CGC Public Apps Gallery, and you will hear from some of the tools for ATAC and Chipseq analysis from her. The second session will start at roughly about 1 p.m. Eastern, and it's titled the SBG Image Processing Toolkit, EC Image Classification with no coding needed. See how we're trying to make it easy for everybody. This session will be led by Soner Koch and Anna Stankovic. Soner is a genomic scientist who's actively developing tools and workflows for the PDXNet consortium, and he's also deeply involved in the Seven Bridges image processing as well as machine learning efforts. Anna Stankovic is a bioinformatics analyst. She found the perfect balance between programming and biology in developing bioinformatics tools and workflows. So for the past several months, she led the development of new tools and workflows, which led to the expansion of the Seven Bridges image processing toolkit. So you will hear about all the new tools that are made available in the Public Apps Gallery. And the last session will begin at about 1:45 p.m. Eastern. The title of this session is Using the Power of Cloud Computing to Scale Single Cell Analysis with the CGC. So it's an exciting session all about single cells. This session will be led by Dr. Manisha Ray and Dalibor Velkovic. Manisha is the program manager of the Cancer Genomics Cloud at Seven Bridges. She leads the entire life cycle of the CGC program and also leads the outreach component of public programs within Seven Bridges. Dalibor is a bioinformatics analyst as well. And as a bioinformatician at Seven Bridges, Dalibor focuses on tools and workflows for, you guessed it, single cell analysis. So without much further ado, I'm going to pass over the mic to Dr. Jeffrey Grover. Jeff, do you want to take away and start the session?
Yes, I can get started now. Can everybody hear me okay? Is my screen showing up? I'm assuming yes, since we did practice this.
Great.
So Thanks for that great introduction, Sai. As she said, my name is Jeff Grover. I'm a genomics scientist here at Seven Bridges. And along with my colleague Nevena, we're going to be discussing today how you can use the Cancer Genomics Cloud to scale up large epigenomics experiments, as well as a bit on how you can use multiple omics data types together so that you can generate more comprehensive biological insights. And so just to get started, a brief outline of what we're going to be discussing today. I'm going to start off with a quick introduction to the Cancer Genomics Cloud and some of its features, then moving into epigenomics at a glance, a deeper dive into two recently added public workflows for Ataxic and Chipseq, as well as how you can use the CGC to do multi-omics analysis. And then I'm going to hand things over to Nevena for a few more details about those workflows, as well as a real-world example of combining multi-omics data sets together. And so first, we should start with what is the Cancer Genomics Cloud. So the CGC is a stable and secure, highly customizable cloud storage and computing platform. It's very user-friendly, allows you to do collaborative analysis of on petabytes of public data, alongside your private data that you might want to bring onto the cloud as well. It's an optimized venue for reproducible analysis using validated bioinformatic tools and pipelines, and so it provides easy management of easy management of your data, as well as scalable computation, optimized bioinformatics algorithms and tools, as well as it's a secure platform where you can collaborate. It's very flexible, and you have lots of confidence in the reproducibility of your methods. In addition to being very user-friendly for the non-coders out there, it's still extensible and developer-friendly for the expert developer or bioinformaticians in the audience. The CGC can connect you to a plethora of multi-omics data sets. And the really nice thing about that is that you can operate on them in place, already in the cloud. You don't have to spend time and money to download them or to upload them anywhere. And you can also use or create reproducible workflows using the common workflow language, and all of our tools are implemented with described in that, as well as Docker. We also provide over 400 optimized tools and workflows with which to construct your analysis in our public apps gallery, but you can also import them from external repositories, or you can write them yourself, either writing the actual CWL code like in a text editor, or you can use our graphical editor you can see here on the bottom right. In addition to workflows, we also have the capability to run computational notebooks through our DataCruncher feature. And these allow you to run RStudio and Jupyter Lab notebooks, and again, operate on your data in place without the need to spend time and money downloading it or re-uploading it again. And you can use this on the outputs of your workflows so that you can refine hypotheses and then also test those. So overall, the CGC has a lot of advantages. You get seamless access to high quality omics data, spend time and money downloading or transferring it, a selection of public and curated workflows and tools available in our public apps gallery. You can organize your data into projects and share that with collaborators. Of course, you can keep that private if you wish. It's also very flexible, and you can bring your own data or your favorite tools and explore those using computational notebooks. And since it's powered by the cloud, you get computational resources on demand whenever you need them. Now, shifting gears a bit, I'm now going to move into a discussion of a brief description of epigenetics, or as you'll see, epigenomics. And so you might want to first define it. And you should probably start with epigenetics rather than epigenomics. And what you'll quickly find if you start looking through the literature, there are lots of diverse definitions that you can find, some of which don't really describe the variety of research done in the field. But what I think most in the field will agree with is this definition from the NIH Roadmap Epigenomics Project. It describes it well in my opinion. And that would be heritable changes in gene activity and expression, but also stable long-term alterations in the transcriptional potential of a cell that are not necessarily heritable. So it's not necessarily requiring heritability, just something that affects the capability for regions of the genome to be transcribed. And when you scale that up to discussion of epigenetic phenomena on a genome-wide scale, that's when you move from epigenetics into epigenomics. And you can see here that with your genome plus your epigenome, that explains a large component of the expressed regions of the genome, mostly genes, but of course also other areas as well. And so when discussing epigenetics or indeed epigenomics, you're frequently most interested in epigenetic modifications. And so these are modifications to DNA and chromatin that affect gene expression, but without corresponding changes in the actual DNA sequence. And some of these might be DNA methylation, as you can see here, as well as histone modifications like acetylation and histone methylation, for example. And then you might also be interested in, say, RNA interference. And in that case, you'd be interested in microRNAs or some other non-coding RNAs. And using specialized modifications to next-gen sequencing protocols, you can detect these changes. For example, if you were wanting to study DNA methylation, you might use bisulfite sequencing. If you wanted to study RNA interference, you would use small RNA sequencing. Likewise, if you wanted to study the three-dimensional conformation of chromatin within the nucleus, you might use Hi-C. And of course, the two workflows that we're going to discuss most through the rest of this, Chipseq, if you wanted to, say, study proteins that interacted with chromatin or modified histones themselves, as well as Ataxic, which can show you regions of chromatin accessibility genome-wide. And what also is apparent, if you look through the literature, or you've performed any experiments like these yourself, there are some challenges associated with doing epigenomics as far as like data analysis goes. First of all, the library preparation methods can sometimes be a bit more complicated than something like an RNA-seq. They're also potentially destructive to the source material. So this might lead to an increased need for read depth or a larger. It also leads to the possibility of bias, and so that's a significant problem in like reproducibility. There's also a lack of consensus between analysis methods. If you look through the literature, if you look at, say, like a DNA methylation experiment, if you look at 3 different papers, you might find similar things, but using very, very different tools. Those tools also might make some assumptions in the way that they're coded, and those may or may not be documented. And then the downstream analysis after you've run your workflow can be sometimes very bespoke and very specific to the actual data set that's being analyzed. And so this leads to a problem in not knowing necessarily what the best practices are. And this also leads to a need for large amounts of storage to handle the large data sets that you're working with. You ideally would like things to be automated, since these can be rather long-running workflows that you need to work with, and you don't want to have to keep track of all of the settings for every single tool. You'd ideally like to have many parallel compute nodes so that you can complete a large experiment in a reasonable amount of time. You also want to be able to share your results and your data without needing people to spend hours or even days potentially like downloading that data. And then you always want to have confidence in the reproducibility of your results. And all of this leads to the CGC being a great environment in which to complete this sort of work. So actually within the public apps gallery on the CGC, we have several options for epigenomics analysis. If you're working with DNA methylation or small RNAs, we have tools for that, as well as our 2 most recent additions to the public apps gallery for Ataxic and Chipseq. And so now I'm going to discuss a bit more in detail about those two workflows in specific. So the first of these is Ataxic, or assay for transposase accessible chromatin with sequencing. I'm going to stick to Ataxic for the rest of this since that's a mouthful. This is a method that you can use to map chromatin accessibility genome-wide. As you can see here on the top right, that you can use a transposase to insert sequencing adapters into regions of open chromatin, but in regions where the nucleosomes are tightly packed, you end up not inserting those sequencing adapters. And once you sequence that, your result is that you get increased read depth in regions that have accessible chromatin. And in general, chromatin accessibility is associated with expressed genes, or at least genes that have the potential to be expressed. Additionally, disease states might exhibit unique signatures or changes versus a normal sample. For example, cancer, when it comes to their chromatin accessibility profile. And in general, it's not too hard to understand the workflow that you have to go through to be able to analyze this data. You start with adapter trimming, alignment to a reference genome, removal of PCR duplicates. These are all fairly standard steps that you would use in many different types of omics experiments, but then you have to move on to peak calling based on the read depth throughout the genome. And of course, there are many options of programs you can use for that step, and they do change the results quite a bit depending on which one you want to use. So this leads to sort of a paradox of choice situation, and of course, the lack of knowledge in the field about what is a standard, right? So in order to help you with that, we've actually implemented one of the most well-used workflows for Ataxic from the ENCODE consortium. Now you'll see here in this view of the workflow, this is quite a bit more complicated than the simple 4 steps that I explained earlier. And that's because this can handle replicate data. It can also give you quality control reports so that you have confidence in the reproducibility. And every step exports logs that you can back check. And just in general, it's a very well-developed pipeline that's been used thousands and thousands of times. And our bioinformatics team here at Seven Bridges did an awesome job working with this workflow to make sure that it executes well and that it's implemented in CWL. So even though this is very complicated looking, you can actually run it very easily since we've already done all the heavy lifting for you. If you are on the public apps gallery, all you have to do is click the run button, basically, as long as you have the right input data. So it looks a little scary, but actually in practice, you don't have to do most of the heavy lifting here. Similarly, we've also implemented a Chipseq workflow, and so that's chromatin immunoprecipitation followed by high throughput sequencing. So Chipseq you can use to identify interactions of chromatin-associated proteins genome-wide. And so your target for this might be a transcription factor, it might be chromatin remodelers, or it might be modified histones themselves, whether they're acetylated, methylated, ubiquitinated, etc. And so after you precipitate your protein, you then sequence the associated DNA with that. And this gives you increased read depth in regions where there are these protein DNA interactions. And so just like Ataxic, Chipseq signatures might identify a disease state or be, say, different between different time points or passages of cell lines. The actual general workflow is very similar to Ataxic. And just like with Ataxic, we've implemented the ENCODE Consortium's Chipseq workflow as a public app on the CGC. And just like with that workflow, our internal bioinformatics team did a great job making this easy to run. So all you need to do, again, is find it in the public apps gallery and click run. So once you've completed your workflow runs and you have some great data, that's great, but you've really taken an input file that's a text file and you've output a text file, right? So you want to be able to get biological insights from that data. And one of the best ways to do that is to actually use multiple omics data types together. And the CGC is, again, a great place for that. So for example, you might have RNA-seq data and you might have Ataxic data. And the RNA-seq data might be between two disease states, between normal and tumor, et cetera. You might have the same sample types for your Ataxic, and that will then let you be able to stratify your differentially expressed genes based upon Ataxic peaks, whether they're showing increased chromatin accessibility in one of the two samples that you have. You can also use this with Chipseq and ask if regions that show changes in the histone modification landscape, are those showing similar changes in the RNA expression profile in those same regions. You could also combine this with whole genome bisulfite sequencing and ask, for example, oh, I have increased or decreased DNA methylation in a promoter or a gene body, or perhaps some trans region elsewhere in the genome. Are those associated with increased expression of various genes or regions of interest? And this doesn't have to stop at RNA-seq. You could use variant calling data, say SNPs, structural variants, indels, and ask whether those have an impact on, say, the DNA methylation landscape, or perhaps the general chromatin accessibility state. And so there are several options for this sort of analysis on the CGC. If you want to do quick exploratory work, but you also don't want to have to spend time downloading your data set, if it's huge data, you can use DataCruncher, and you can do interactive analysis there in Jupyter or RStudio notebooks. If this is an analysis that you're going to do many times, and let's say you've figured out what your steps are going to be, you can implement this as a CWL workflow, and you can run this at huge scale and optimize for runtime and for, of course, computational efficiency. And so now I'm actually going to hand this over to Nevena, who's going to show you a real-world example of how you can use multi-omics data sets to be able to get some really perhaps interesting biological insights from your data. So you can take it away, Nevena.
Thank you, Jeffrey. So can you see my screen? I suppose yes. So thanks again. I'm going to walk you through.
We're actually seeing you, not your screen.
Really? Oh, okay, sorry. I'm trying it again.
There we are. I think maybe I just didn't close mine soon enough.
Yeah, probably. Okay. Everything cool now? Yeah. Okay, so I'm going to walk you through an example of a multi-omics analysis that combines epigenomics and genomics data. I'm going to show you the design of the pipeline that we made and give you a quick demo on how to run this pipeline by yourself on the CGC platform. So we wanted to make a pipeline that would combine RNA-seq data with Ataxic or Chipseq data in order to explore the correlation between chromatin accessibility or histone modifications that can be indicators of active chromatin sites with gene expression. And this is how the pipeline design looks like. As you can see here, it has three main functional parts. There is this yellow RNA-seq branch. There is the blue Ataxic or Chipseq data processing branch. And there is this green part that actually combines these two data types together. So let's start from the beginning. So first, we are starting from RNA-seq reads in FASTQ format. And the first step is initial processing of RNA-seq data that involves a QC step, which is done with the FastQC tool, and alignment and quantification step that is done with a Salmon tool. On the other hand, we are processing Ataxic or Chipseq data, also starting from FastQ files and Ataxic or Chipseq reads. And the initial processing is done using the before mentioned encode pipelines, which, as Jeffrey said, involve QC step, alignment step, and peak calling step. And this part of the pipeline can be done using our CWL workflows that are publicly available on our platform. And now I'm going to show you how that looks like. So this is my project, for example, and I'm going to go to the tasks page here, where I already prepared these 3 draft tasks, which we are going to run now. So first we are going to run the FastQC analysis, which is the quality check analysis for our RNA-seq data. This is a pretty simple tool. So all we have to do is just click here, our RNA-seq samples, save them, and just click run, and our task is starting. And the other task is the Salmon workflow. We are going to align and do the quantification of our RNA-seq reads. So here we are doing that with Salmon workflow. So here I already added all the reference files. We need GTF annotation, genome FASTA, and transcript FASTA. And we are also going to add RNA-seq data. We save them. Then all the inputs are prepared. And here we have different parameters that can be changed. I'm going to leave everything as defaults for now, but for example, let's say I want Salmon to perform GC bias correction, then I'm going to set this parameter to true and just run the task. And that's also done. And the last one will be encode the Ataxic pipeline. So now we are processing our Ataxic files. This is very similar to the Chipseq data. So I'm just going to show you this example. As you can see here, I also prepared and added all the reference files because there are a lot of them. But they can be easily downloaded from the GitHub repository page of the attack encode the Ataxic pipeline. So all we have to do here is to add our input files. I have a paired end sample, so I'm going to add read number 1 here. So I have here these 2 samples which belong to Ataxic, but first I'm just going to check if all the metadata fields are set accordingly. And yeah, there are, like I have paired end set to read number 1, and I have case ID and sample ID also set, which is important because this workflow knows how many technical or biological replicates do we have based on those metadata fields. So, okay, I checked, everything is fine. And I'm adding this read number 1 here, and also the second read here. Yeah, okay, I'm going to also run this with the default settings, and I'm just going to add the required settings here. This one is required. It's max 2 genome sizes. I can see here in the description that for human samples, I should type in HS, and I have human samples, so I'm typing that. And here I will set the paired end parameter to true because I have a paired end sample. Everything else I would leave like this and just run the task. So now all my tasks are running and this part of the pipeline is done. Of course, these tasks are going to run for some time, but I already have the same task completed. So I'm just going to quickly show you how the completed tasks look like. This is a completed FastQC task. It's very simple, as you can see, and we have on the outputs reports in HTML and zip format. We can just have a quick look. So this is a FastQC report, as you probably know. Then we have the Salmon workflow, a completed task. We can see here that the workflow lasted for 30 minutes and the price was about $50. And we have all the outputs. We have gene level and transcript level quantification files. And we have expression matrix for genes and transcripts, which can be useful if you have more than one sample on input, and some log files. but this is the file transcript level quantification that we are going to need for the further analysis. And the last task is encode the Ataxic pipeline task. This is the complicated one that Jeffrey showed you. And we can see here that it lasted for 9 hours. The price was something about $4.50. And we also have a lot of outputs here. What is important for us now is the alignment files, BOM file. We also have the filtered BOM if we want to use it. We have the cold peak file in narrow peak format. And we also have the QC report if we want to use it. So that's the first part of the pipeline, and it's done. So now we're going back to the presentation. Let's just do like this. Okay, so we finished this part of the pipeline, and now we have the quantification files from RNA-seq step. With these files, we are going to do the next step, differential expression analysis, which is done with the DSeq2R package. And on the other hand, with cold peaks that we've got and alignment files, we are going to do a differential binding analysis for the peaks using the diffbind-R package. And we are going to annotate those peaks using the Chipseq-R package. Then we are basically going to have 2 lists. So we're going to have a list of differentially expressed genes, and we're going to have a list of differentially bound peaks, which are going to be annotated by the nearest genes. And then we can combine those 2 lists, overlap them, and filter only the differentially expressed genes that are also present in the differentially bound peaks file. And with that filtered list of genes, we can run a gene set enrichment analysis using the GCA function from cluster profiler R package. So the main outputs of the whole pipelines are going to be a list of annotated peaks, a list of differentially expressed genes, overlapped list of genes, and the HTML report that I'm going to show you in a few moments. And this part of the analysis is done in RStudio in DataCruncher, and it is available as a public interactive analysis, which can be found here. Just, here, sorry. So you can go to the public projects menu, then choose the DataCruncher interactive analysis project, go to the project, choose the analysis, is the last on the list, called Epigene Multipleomics analysis, and run it, and click on it, and then copy the analysis to your project, and you can run it with your own files. Before I show you the analysis, just want to mention that for the purpose of this example and for this public interactive analysis, we use the data sets coming from this paper from Tsai et al. They investigated differences in epigenomics and genomics of this MDA MB231 breast cancer cell line and its brain and lung metastatic subpopulations. And we used their 2 data sets, Ataxic data set that consists of 6 samples. So there are 3 cell lines represented with 2 replicates. And we used the RNA-seq data set with 9 samples, so 3 cell lines and 3 replicates. And we integrated these 2 data sets in our multi-omics interactive analysis, which is something that I'm going to show you now. So I'm back on the platform. And now I'm going to go to this DataCruncher tab. This is my copy of the Epigene Multipleomics analysis. It's already running, so I'm just going to open it. And here I have this R notebook file with the code. I'm not going to go through the code now, but just want to mention that it's very easy to find the places in the code where you can just change the code so you can add the paths to your own files or folders and then modify the analysis and use it for your own data. but I'm going to walk you through this HTML report that is the result of this analysis. And let's just have a quick look at it. So it has different sections. The first part is Ataxic or Chipseq downstream analysis, starting with the differential binding analysis. Here we are loading the metadata. We have condition, which is going to be the design formula for our differential analysis. We are counting the number of peaks in each peak sets. We can make this correlation heat map, which gives us an initial clustering of the samples. I'll look at it. Then we are using the BAM files and computing count information for each of the peaks and regions. And with that information, we can also make some, again, some correlation heat maps or make a PCA plot and have a look how our samples are clustered together based on the condition that we chose. Then comes the differential binding step of the analysis. As I said, we chose the condition as a design formula. So we have basically 3 groups that we are going to test. Because DiffBind uses the DSeq2 algorithm, we can see here the number of differential peaks after the DSeq2 analysis. We can also visualize the result of the analysis with many different plots that we want. And after that, we are annotating our peaks. And also we can visualize this by making these feature distribution plots like this. Or if we want to show multiple groups on the same graph, it can be done with these plots. Then the second part of the analysis is RNA-seq downstream analysis. So now here we have differential expression analysis of RNA-seq data, and it's similar. So we have also loading the metadata. Now here we have sample type, which is the same, the condition, the same design formula. First, we can do this quickly exploratory analysis, also make a PCA plot, see how our samples cluster. Then the differential expression analysis itself. We have the same groups here that we test, and we have a list of significant differentially expressed genes that are up or down regulated. And we can see them here in a table, or we can plot them in a volcano plot and visualize them like this, for example. And the last part of the analysis is where we are combining actually those 2 lists and filtering the genes that are present also in the differential bound peaks file. And with that list, we are doing the gene set enrichment analysis. And the output of this analysis is a table like this with gene sets that are enriched, their significance, enrichment scores, and so on. And we can also visualize this with a dot plot like this one. So that was just a quick look at the HTML report. But as I said, you can very easily change the code and make yourself a report like this for your own data. And with that, I would like to conclude with some key points. To repeat that the CGC allows you to seamlessly access petabytes of epigenomic and multiomics data, that using the platform, you can scale your analysis and run large-scale epigenomics experiments. The platform is flexible. It allows you to bring your own workflows, tools, and data. And by using it, you can combine multiomics data to generate more comprehensive biological insights. And with that, I would like to thank you for your attention. And if you have any questions, please don't hesitate to ask. Thanks.
Great. Thank you so much, Jeff and Nevena, for this very insightful session on epigenomics data analysis. And I'm really amazed at how complicated that workflow is, but how you have made it simple for the researchers and, give them more time to focus on the science. So we are happy to take questions. So please feel free to type in the chat. We do have a few that have come in. So I'm going to start reading them out. And I know our team has been really prompt with responding to questions in the chat, but I'm just going to, call out so that everyone is aware of the questions and the answers we gave. So the first one we got was, is Linux used for writing algorithms? And also, do we have Jupyter, primarily Python and R? So I've provided the answer, but Jeff or Nevin, do you want to take one if you want to respond to that?
Sure, I can answer that, I believe. Yeah, so we have both RStudio and JupyterLab available through DataCruncher, but in even in the JupyterLab sessions, you can use the R kernel there. I frequently actually prefer that myself. I like the JupyterLab interface better, but I mostly use R for exploratory data analysis. So yeah, that works pretty seamlessly. And as far as Linux goes, yes, the containers that run all of the tools, I believe, are at least mostly based on Ubuntu. So Linux is at the core of what runs on the back end.
Great, thanks, Jeff. Okay, so we have one more question, which was, does CGC have a workflow for bisulfite sequencing for DNA methylation? And I know, Jeff, you have posted links to the public apps gallery and the tools that are available. Do you want to add anything?
Yeah, so actually, this is a great question for me because that's exactly what my background is in before coming to Seven Bridges. So we have two workflows, the Bismarck workflow, which is pretty much the standard, and then also a workflow using Bitmapper BS, which is a little bit faster, newer tool, as well as MEthylDACL for methylation calling, which I've used in the past as well. And yeah, these are all available on the public apps gallery with, of course, benchmarking information as well. So you can have a really good sense of what your cost might be based.
Great. Nevena, do you have anything to add there? I don't know, Jeff, are you still talking?
No, I'm not still.
Okay, great. Sorry. Nevena, do you have anything to add about the other kinds of epigenomics tools?
Well, I actually wanted to answer this other question. I don't know if you...
Sure.
Yeah. There was a question about the size of input files for the workflow, right? Yeah, so the size for the RNA-seq data, this sample was a paired-end sample, and each of the file had around 5 gigabytes. And for the Ataxic files, they were a lot bigger, each of them was around 18 gigabytes. You also asked about the price of the task. Are there tables? So yeah, there are. For every workflow, when you click on the workflow page or a tool page, you will have a description of the workflow and the table of which is called performance benchmarking, which we did, and there you can find all the informations. How much the cost of the task will be based on the input file sizes.
Great, thanks, Nevena. Okay, so we had one more comment and question. Price is available on job completion, that's great. So yes, we do provide the price and the time taken. And actually there are more details, like what was the instance size that was used, all those kinds of details are customizable, and you can also see them and tweak based on your sample size and needs of your experiments and analysis. So the question was, are there tools or tables to help with cost estimation? So Jeff pointed out that yes, there are some tools for benchmarking, and Nevena also mentioned that. Does anyone want to add to that, Jeff or Nevena?
We've also worked on some sort of general guidelines for cost estimation in the cloud. So I don't know exactly when that, that's going to be written up as a public tutorial that users can access CGC like documentation, as well as our other platforms. So I don't know exactly when that's like going live, but I would recommend if you have a specific question about it to reach out to support. We can usually help you specific questions like that.
Absolutely. Thanks, Jeff. And I think that's going to happen. The tutorial is going to go live very soon. So keep an eye out for that. And also, if you sign up for our newsletters, you will see these notifications about new tutorials. We want to go to the next question from Vincent. Is this a FISMA moderate security environment? So I can actually answer that. Yes, we are a FISMA mod, and we'll likely be FedRAMP certified as well very soon. So if you have more questions, there's a detailed documentation page in the CGC website about our certifications and the security features. And of course, you can always reach out to us directly, and we'll give you more details. And okay. The next question is, so Nevena already answered the size of the data that was used. We have one question. I'm wondering if the jobs fail to execute, is that possible to investigate the reason why it failed internally?
Yes. So actually, as you might know, if you've run a lot of bioinformatics tools, sometimes things don't go as planned, right? So we actually capture logs and like command line output to standard error from each of the steps in a workflow. If something fails, you can go into those logs just as you would if you were running it on a local machine. You can also view real-time stats for each of the like instances as they run, which can also give you hints. For example, if you had one file out of a large batch fail, you find out that particular file, the instance that it ran on, ran out of disk space or RAM. That'd be a pretty good clue that you need to run it again with more resources. If something like this happens, we also have features like memoization, where if various portions of the workflow worked fine before a certain step and then it failed there, you can pick up the analysis from where it last was successful. That way you don't have to repeat steps that actually worked just fine.
Okay, great. All right. One more question we have is, how much did the demo analysis cost? So trying to get an idea. So I think, Devena, you showed it, but you may want to just provide a more.
Yeah, it's very difficult to say exactly. I mean, what I've showed you now is just an example, a task example with one of the samples for the workflows. But when I did the whole analysis, I'm just thinking I'm assuming it depends on the number of your files and their sizes. But I'm assuming that this analysis that I've just showed you was everything together, maybe less than $50. So that's what I would say.
Great. Yeah, and the platform also has, at every stage you can look at the cost in the task page, like Nevena had pointed out. Great. Do we have any more questions for Jeff and Nevena? All right, so we are a little bit ahead of time. So we will move on to the next section, which is going to be image processing and machine learning on the Cancer Genomics Cloud. And you don't have to know any coding. So that's the title of the topic. So I'm going to hand over to Sonar and Anna. You can get started, Sonar. We can see your screen. Maybe you're muted.
Yeah, now you can see me and you can hear me, right? Okay, perfect. All right, yeah. Hello, everyone. I'm Sonar. I'm working as a genomic data scientist at 7 Bridges today. And then I will talk about the power of AI on the Cancer Genomics Cloud and our recently published image processing toolkit. Yeah, hopefully it will be helpful to everybody. Let's get started. Before getting started, this is our agenda for today. First, we will see how easy to do AI on CGC, and I will present our image processing tool along with some cool demo. Then we will be happy to get your questions. Let's talk about what CGC offers for machine learning and deep learning tasks to users. Before going to more AI-specific steps, I will briefly mention the key parts. In the next couple of slides, we will see the meaning behind these with some examples. CGC offers seamless integration of data, code base, your AI model, computation environment for reproducible pipelines with great compute power. It helps bridging the gap between the exploratory computing environment, production application, really easy and user-friendly way. It's highly secure, has powerful robust documentation to start quickly, and if needed any help, we have team who experience in the field. Let's go into more specific needs. What are the key points of AI project from data exploration, model engineering to production? All listed here, and CGC has great features for each of these. First, ETL, data readiness for AI is one of the most important and time-taking step in the AI projects. And feature engineering, especially for traditional machine learning models, model selection. It's always great to have models ready to use or train from scratch or prefer transfer learning. And hyperparameter tuning, which is the problem of choosing the set of optimal configurations for a learning algorithm, like learning rate, depth of network, etc. Model training comes one of the most fundamental one, training your model with desirable frameworks. Then prediction comes to observe the performance with unseen data with nice visualization, and then experiment logging to track how your model behaves and learns, and also important for reproducibility. And at the end, the clean and understandable analysis results in shareable format is required for interpretability. What I would like to say, all these are smoothly integrated within the CGC. We will see the details in the next slides and during the demo as well. This is the whole view of machine learning lifecycle on the CGC platform. As you can see, it has 6 main steps. It starts with data integration. You can bring any public or private data you have, or you can access popular data sets directly from the CGC like TCGI, TCIA. As all we know, everyone wants the clean data and don't want to clean the data, but generally we need to do some cleaning and preparation. Data readiness for AI is really important since the data is food for AI. CGC has great features for it as well. You can do all your ETLs in your project on the CGC. And for core machine learning steps like feature extraction, model engineering, CGC also offers great features that make your life easier. You can easily set up an environment with any framework you want, TensorFlow, PyTorch, Keras, etc. With DataCruncher feature, which is an interactive analysis tool on the CGC platform using the Jupyter Lab. It's completely integrated to the platform. And to find the best model, it's always necessary to tune the hyperparameters, configurations of a selected model. With the power of multiple GPUs on the CGC, each configuration will run in parallel with batch processing. And at the end, it will return the best trained model and its configuration to you according to the pre-selected performance metrics. All these are tunable and can be specific to your project and learning model, of course. And integration of your code base is also straightforward. You can clone your code base from your repository, or you can directly upload it and immediately start to work on it. And when you are done with your pre-processing and modeling, evaluation of your learning model comes. This is also a very important step for AI application development lifecycle, since evaluation will tell you how successful you are in your data readiness and your model engineering step. Maybe it's overfitting, underfitting, but tell all this to you. In addition, CGC offers nice monitoring feature and visualization capabilities. User can monitor, visualize, and diagnose the issues if there's any. And at the end, after considerable amount of idea, experiment, and design cycles, when user decide that this model performs well, The next step will be to serve this is a tool or notebook or reproducible workflow, which is one of the most powerful side of the 7Bridges platforms that help you to make your project portable, readable, and replicable. You can share your model as a common workflow language with your collaborators. We also have Onyx support. Developers are often logged into one AI framework or ecosystem. Onyx is enabling more of these tools to work together by allowing them to share models. User can export its model to Onyx format, then deploy in much supported machines. Yeah. As I briefly mentioned, DataCruncher is an interactive analysis tool on CGC for data exploration, model engineering, and visualization using familiar frameworks like JupyterLab and RStudio. It helps fast prototyping, actually. You can start to work on your data with your desired model in a minute and evolve from prototyping to production-ready tool chain. A couple of key features of DataCruncher are from ML. It allows you to perform your ML analysis directly where your data is. It allows you to save outputs of model and prediction to your project on CGC. Use JupyterLab or RStudio as a computational environment. Of course, configuration is possible. You can install any specific environment on it, specific framework versions, etc. It's tightly integrated to the CGC, easy to access to any data on CGC, and share the outputs across the projects over the CGC or download to your log. And yeah, this is more concrete example for fast prototyping your machine learning project on CGC. Show how easy to prototype. We use this flow in Precision FDA Brain Cancer Predictive Modeling Biomarker Challenge, which is ranked as a second place solution by Predictive Performance. Briefly identifies biomarkers and predict the patient outcome using gene expression, DNA copy, and clinical data. Here, the first data is transferred from the challenge resources, which includes the multi-omic data set. Then data repository is cloned to the created DataCruncher instance. You can install your environment in there, make necessary changes, maybe some fixes, testing, then execute the model and visualize the results. You just in one place, everything you can do. You could probably use this for most of the machine learning or deep learning workflows. This fast prototyping also increases the user flexibility. If user plan to edit, update the ML analysis pipeline frequently, easy to do lots of back and forth with that one. For example, there can be a couple of reasons. These are the most common scenarios. It can be data-derived. The model was trained on a certain distribution of inputs, but the distribution changed over time. It can be a concept-derived. The model was trained to learn the X to Y mapping, but the statistical relationship changed between these variables in time, or can be a changing requirements, like the model was built to perform particular tasks, but the product team decided to modify it for different capabilities. And yeah, now how your ML tool is executed when wrapped as a tool or workflow on CGC. First, scheduler assign an empty instance to this job according to the resource description in CWL. Then executor mounts the input files to the instance and pulls the image from the SP Docker repository. You can push all your images to this repository, then create a container. Executor generates comments and run the job. And last but not least, when the job is done, all the outputs you have will be saved back to the storage in your project. Yeah, this is also important. CGC also offers easy to use monitoring and debugging features. User can access the details of instance, cloud type, status of your analysis, instance configurations, GPUs, memory disk space uses, etc. User can access the live logs, standard outputs, errors, warnings, workflow details. More AI specifically, you can track, for example, current epoch number, current training and validation accuracy, it will be sign of overfitting or underfitting, etc. And as all we know, reproducibility and replicability is one of the important blocker in science and also in machine learning for healthcare fields as well. The focus of machine learning community has shifted away from the factors like explainability and reproducibility towards matters of raw performance as measured by evaluation metrics. As a result, many machine learning models are either not reproducible or are difficult to reproduce, even if retraining an existing model with the same hyperparameters, configurations, and the data set does not always produce the same result. However, CGC offers the compact cloud environment that you can solve or improve these reproducibility and replicability issues in terms of technically, statistically, and conceptually. Yeah, hopefully all this helped you to understand how useful and powerful CGC is for AI. Now, my colleague Anna will talk about our recently published image processing toolkit.
Hello, everyone. Sonia, if you can stop sharing so I can try.
Hold on a second.
So, do you see my presentation?
Yes.
Yeah. Great, thank you. Thank you, Sonia. So as he said, my name is Anna Stankovic, and today I will be showing you the tools that we have developed and that are already available on the CGC Public Apps Gallery that showcase these possibilities of machine learning and image processing on the cloud. So we have developed and wrapped a set of tools with the goal to enable researchers to create and easily use machine learning image classifiers without performing any coding themselves. So these tools work together to create an accessible, modifiable, and more than anything, easy to use infrastructure that enables various stages of machine learning image processing, like image pre-processing, model training, and image prediction. Some of the highlights we want to emphasize is that these tools utilize platform computing, which brings massive speed and price improvements compared to manual writing and running model training scripts. They also support a variety of medical image types like histology images, X-ray and MRI images, or more commonly used picture formats like JPEG or PNG. Also, with data capabilities of the CGC, this toolkit allows image processing and genomic data integration. So for the tools we have published 7 new tools, 6 of which are developed by the 7 Bridges bioinformatician, and one HistoQC tool is an open source tool. We have 2 custom deep learning apps, HistoQC for quality control. We have several histology or X-ray image pre-processing tools, and 2 utility tools for all of these to be easier to use. Before we look at the tools themselves more closely, here we can see a high-level overview how these tools connect. So starting point is your image dataset on which you can choose to perform before model training a series of QC or pre-processing steps. Then you will be using the main deep learning exploratory workflow. It is used to build and train multiple models on the given dataset, generating comprehensive reports on each one to help you determine the best model for your dataset. And then you will use that model configuration in the final phase, the prediction of unlabeled image dataset. If we look at the QC and pre-processing tools, for histology images, we have published 3 new tools, which are QC, as I said, for quality control. We have SBG whole slide image pre-processing tool, which can help you prepare your whole slide images by removing background, pen marks, or other artifacts, giving you a cropped sections of the images, the tiles that contain only tissue. Also, you can perform stain normalization on your histology images based on a target image with the SBG stain normalization tool. And for X-ray images, we have one workflow which allows you to perform one of several different algorithms for your images. Our main focus of this toolkit are the deep learning apps. These are the central part as they allow easy building and training of image classifiers. And as I said, prior experience in machine learning or even coding in general is not required. Deep learning is actually a subfield of machine learning, and it typically needs less human intervention, but requires more computing power and training data than machine learning. But this is what makes it, using it on the cloud, really ideal. These apps are using Keras, an open source deep API written in Python, running on top of machine learning platform TensorFlow. These tools rely on the transfer learning approach, which means we use pre-trained models from Keras. Those are the models were trained on a large benchmark dataset and published. And then we are using them as the start point to create models that are adjusted for our own image dataset set as input. So these tools this approach allows us to build tools that can be used by a larger number of researchers for a variety of use cases or analyses. There are two deep learning apps that we have published, the SBG Deep Learning Image Classification Exploratory Workflow and the SBG Deep Learning Prediction. So let's take a look at our exploratory workflow. This is the main app in our toolkit. It takes advantage of all the benefits of cloud computing. If we look at the steps, step one is the first tool in the pipeline, the SBG Create Configuration JSON tool. By selecting here various pre-trained models and or hyperparameter values, this tool defines those series of model configurations and outputs them in JSON format. Step two gives us support of various image formats, and step three is then the image processing tool itself. This tool, in combination with those multiple configurations, provides the main feature of this workflow, which is parallelization of the model training process across several different GPU instances. This is what provides that speed and price improvement and ease of use in model training scenarios. The step four are several tools that allows us to prepare those reports for us to be able to see the results of our model training process. As input, this pipeline takes your labeled image dataset organized into train and test folders, and an output you get reports and visualizations, which we will take a closer look after, but this is our interactive HTML report. Here we can take a closer look at the results of each of the trained models and select the one we want to use in our final prediction of unlabeled images. Each model is defined by the unique configuration string, which you can see here. This configuration string is built from all the parameter values that define it. You can copy the string and use it as the input in our final tool in this tool, Deep Predict. This tool is the final app in the toolkit, as it's an image classifier that composes and trains models on all labeled data we have in order to be able to best classify unlabeled images that were included in the validation set. It takes the best model configuration provided by exploratory workflow in the form of configuration strings that we saw earlier. And then the trained model is generated and used to classify unlabeled images. So let's see how these tools look on the platform itself. Set up an example project. We can perform then exploratory imitation with our workflow and then perform image label prediction on an unlabeled dataset. So here is our CGC platform. And first, let's set up new projects to perform our image processing. Here we can select a name for our demo project. We can select our billing group, which will select from where we will pay our AWS instances. And the most important thing for these tools is the location. Because of the deep learning tools that they use the GPU instances, which are only available through AWS for now, we have to select the AWS for the location of our project. And we can select spot instances, which reduces the cost of our test. Memoization is also during development always really useful, which retains our intermediate files, and we do not have to rerun whole workflow again. And also you can select network access or if you will have control data. I have already prepared a project for this demo and we can see it now here. First, let's go and what kind of files I have in my project. We can go to this dataset. This dataset is from Kaggle. Kaggle is an online community of data scientists and machine learning practitioners, and it offers a wide variety of open source datasets for machine learning. This one is the breast cancer images on histopathology slides dataset. It contains microscopic biopsy images of benign and malignant breast tumors. There are around 1,600 images that are originally tiled and pre-processed and ready to use. And they're really easy to import to our platform and then use in your experiments and testing. So here we have these images already organized in our test and train directories. And if I open this directory, you can see that my labels are organized in a subdirectory structure. So all of the benign images of the benign samples are in this directory, while malignant are in these. The other way I can organize my labels is have them in the same directory, but we need to have then a CSV file that will contain those labels, like a manifest file. We can see here we have all of these images in PNG formats, and we can see a preview of the images on the platform itself. So let's imagine that I have a collection of labeled images from this dataset, for example, but also I have my own dataset from my samples, which I'm interested if I can automatically determine the cancer type. So in these files here, I have this unlabeled dataset, and I'm interested if I can use these tools to find which of these samples have benign and which ones have malignant tumors. So to do this, I will first use our exploratory workflow. I can find it in the public apps gallery. Here you can search either by typing the name of the tools, or you can explore all apps and find the category that is called imaging. Here in this category, you will find all the newly published tools and all the tools you can use for image processing on the CGC. For now, we will take a look at the deep learning image classification exploratory workflow, and we can copy it to our project. I already have it, so let's see where it is in my Summer Symposium demo. Now we go to the tab called Apps, and we can find our deep learning image classification exploratory workflow here. Here you can find in the description all the information in more detail about this workflow, like common use cases or issues and important notes, also a benchmarking table, image of the pipeline, and also all the inputs, app settings, and outputs. If we go to the editor, here you can see our CGC workflow editor, which is really easy, really intuitive way to edit your workflow. So these gray nodes here are all the inputs of our pipeline, while these nodes are all our outputs. The green nodes are the tools that we are connecting to each other, and we can, by just tracking these lines, we can see easily how our data flows through the pipeline. And it's really easy to modify, to use, and to see in more detail with the double click each of the inputs, parameters, and settings for each of these tools. We can see here that this is the main deep learning image processing tool. And on the step, we can see that it's scattered on this input, which it gets from this tool. That means this tool is scattered across multiple instances by each configuration it gets from this create configuration JSON tool. So let's go and try to run this workflow. As I said, so we have here several different things that we can select. First are the input files for our workflow. Then we have the app settings or the app parameters that we need to set. First, for the test directory, I already have prepared my test and train folders. If I have not prepared them like this, or my data set is in one directory, there is a utility tool that we have called SBG split folders, which can easily, in just one step, do this for you. So we save this selection, select our train directory, and then go to our parameters. There are several different parameters here. These are the hyperparameters for the model itself. You can see more information on each one and the default value if you click on this question mark. But for now, let's focus on the pre-trained models. So as we said, we're using Keras pre-trained models, and here you can select several models you want to choose. For example, we can select VGG. We can select Inception v3 and, for example, ResNet 50. So as we said, these models were trained on a large benchmark dataset published, and use them just as a starting point for a model adjusted for our image dataset. So each of these pre-trained models have their own properties. For example, these models are widely used for transfer learning, both because of their performance, but also because they were examples that introduced specific architectural innovations, namely VGG for consistent and repeating structure, GoogleNet for inception modules, and ResNet for residual models. As I said, there are a lot of hyperparameters here that you can select, and information on all of them you can find in the description or on this question mark here. What we can also, important thing to set is sort by metrics. This metric is what will determine the best model and output in the plots for that model itself. So our value, we have a large number of metrics that we are tracking for each model, and validation AUC is the default one, but I will select it anyhow. Next step is to check our execution settings. Now I have selected only 3 pre-trained models to test, and all the other hyperparameters are on default. But I want to shorten the time of the execution, so I want each of the model to be run on his own instance. Because of that, I will choose parallelization. This is the number of parallel instances that will be run with this task, and I will select 3. For the app default, default instance is P2X large AWS instance, which is a GPU instance, which is more than enough for any of your tasks. But if you want to choose a larger one, and you choose custom instance here, be sure to select one of the P1s available when you scroll down, because these P instances are the ones with the GPU. And if you select some other, your task will fail. And then when I have selected the default instance, but with 3 parallel instances, I can run my task. And while we wait for that to finish, I can show you a finished task. So here we have the same pipeline ran with a different configuration. So I have several dense layers. I wanted to try the dropout layer with or without, etc. In combination here, I have 18 different configurations, and I ran this task on 6 parallel instances. We can see that this task, it lasted about 2.5 hours and cost $13. Most of that 2.5 hours was in model training. We can see that this job here is that deep learning image processing app. And all of these separate green jobs are the single configuration being trained on each of these instances. If I select this, I can go and view logs. So here I see all the jobs for every configuration that was trained. And here I can see the command line that was executed. I can see what was outputted. And this maybe is the most important when developing. This is an error log, which catches all of the training steps that are logged, like, for example, fitting the model, then going through every epoch, and then calculating metrics, plotting, and in the end, a successfully finished job. So we can see that this parallelization is really good. And we can see, if we go back to my presentation, we can see how much time this can save us. For example, if we look at this data set, which has a 620 megabytes in size, if we train one configuration on one parallel instance, it lasts around an hour and costs around $1. But if I want to train 24 different configurations, that would take me a whole day, 24 hours. But by using parallel instances, I can do this in around 6 hours. So I come to work, I run this task, and by the end of my workday, this task is done, and I can already see the results, and it costs $30. If we go back to our task, to our finished task, let's see what results we get. So as I said, our main output is this HTML report. If we look at this report, we can see, first thing we can see is our model metrics. So each of these models, as I said, is identified by this configuration string, his unique idea, which is made from combination of each of hyperparameters that was applied. Table is automatically sorted by the validation AUC, which we selected as input. But if I change my mind and I'm interested in some other, like validation accuracy, here I can also resort my table to see any metric that I want. So I can copy this string for our next tool. Also, because there are a lot of different metrics, for easier use, you can still select which ones you want to see or want to hide. For example, let's say I'm not interested in validation precision AUC, but I want to see validation accuracy, and I select this model to be my chosen model for the final prediction tool. Here in my HTML report, I also have plots, but these plots are based on criteria that we selected on input. So we have confusion matrix, precision plot, loss plot, and AUC plots. You can click on these plots as well if you want to see them in more detail. But let's say I have my mind and I'm interested in validation accuracy. So we selected this model as our best one, and I will copy it. And if I go to my outputs, I can see that all of the positions for all the configurations are also in this zip archive. So even though I change my mind, I can find my plots in this archive, and I do not have to rerun this task and waste another 2 hours. So what can we do with this configuration string? We will now perform our final image prediction on the data set that we have, which is unlabeled. So my own images from a laboratory that I'm not sure to see which type of cancer they are. So if we go to our apps, find there our deep learning prediction tool, and we can then select to run it. Here for inputs, we do not have train or test, but we are selecting labeled images, which are both train and test images. So we want to use all the labeled images we have available. And for unlabeled images, as I said, we have then for the configuration string, I want the value that I got from my HTML report. But also if I, for example, I didn't run my exploratory workflow, I have an idea of which hyperparameters I want to set. I can also set them separately here. But just to mention, here we train only one configuration, so you can only choose, for example, one pre-trained model and not several others. So here we can set our configuration string and then run our task. And while we wait for that, we can show our finished task. So we see here that we have selected the same inputs and a configuration string, and in the end we get this, which is our predictions CSV. It contains image IDs, predicted labels, and the prediction probabilities. These tools also offer support for both binary or multi-label or classification. So if you have multiple classes, they will be also present in the table. But what do we do? Let's go back to our presentation. So what do we do if we already have our own ML script, or we have some repository that we are interested in testing on the CGC? So this is that fast prototyping that Sonia mentioned, and we can see how easily we can port it in form of a tool and integrate it into a workflow. For example, let's look at this repository, which is the NCI DOE Collab Pilot 1 Tumor Classifier repository. TC1 is a one-dimensional convolutional network for classifying RNA-seq gene expression profiles into 18 balanced tumor types, like breast cancer, melanoma, etc. The model is trained and cross-validated on a total of 5,400 RNA-seq profiles from the NCI Genomic Data Commons. And I want to use it on the CGC. Let's go to our tool. So this is a tool that Sonia prepared for us to present how we can easily do this. So as Sonia previously explained, these tools run in Docker containers on the AWS. So you can build your own Docker image containing all the libraries you need and push it to the 7 Bridges Docker repository, and then call on that image in this Docker image field. Then in this base command, in just a few lines of code, we can clone the repository, and it's already ready to run with Conda. Other than that, we set up our inputs and outputs just to prepare it for the final workflow. And let's look how it looks inside our modified exploratory workflow. So we have here tumor classifier workflow. See, so it's really similar. We also have creation of multiple configurations. We also have the creation of the HTML report, but we now do not have input images on our input, but we have RNA-seq data, and now a whole new experiment, really easy in several clicks and small modifications of the published tools. So it's really easy to use, and it's really flexible for any kind of your experiment. And we hope that you will try to do this and try to have fun with these tools and utilize the multiple GPU infrastructure that we have created. So what we have seen that it's really easy to access image processing apps. We can see that these tools enable fast and cheap analysis and image prediction in the newly published apps. So, and also easy integration of custom machine learning tools. For the conclusion and future work, we summarized how powerful CGC is for AI healthcare projects. It's easy to scope AI projects and deploy them on CGC. And image processing toolkit is publicly available, and you can start to use it. Feel free to play with it and share your feedback with us. These tools are all in constant progress, and we are modifying them. So anything you see that you're interested in learning more, have any feedback for us, how we can improve on them, we are really, really eager to hear from you. For the future work, we are planning to extend on this image processing toolkit with more pre-processing and post-processing options for different data types and to make them ready for the AI model. More state-of-the-art models and workflows will come soon for cell segmentation, classification to drug response prediction, and also more end-to-end fully connected and fully convolutional networks with tunable capabilities. We also want to make implementation of multi-omics data with deep learning and multi-omics with imaging deep learning solutions. We already started on that, and we hope to publish them soon. Then I want to acknowledge all of our bioinformatician, our CGC staff that really helped us and supported us through this effort. And thank you very much. These are our contact mails if you have any feedback or questions. Thank you so much.
Wonderful. Thank you so much, Anna and Soner, for the great presentation and live demo. It was very enlightening to learn about the new tools that have been published. And we have a bunch of questions, and I can see there's a lot of interest in imaging. So that's very exciting. So a few have been answered, but I'm just going to call out, and if Anna and Soner, you have anything to add, feel free to answer them. So the first question that came when we started the imaging session was, what is the cost for uploading, downloading private data and for storage in CGC, or where can I find the information? I think Manisha posted the information. Anything to add about that?
That's fine. Yeah.
Okay, great. So we basically pass through the storage charges, which Manisha has mentioned. And of course, there's no cost for uploading, but you load. So that's something you can find more details in the pricing page. The next question is, are the images expected to be de-identified before uploading to the CGC? Anna or Soner?
Yeah, it will be great to elaborate more what you mean by the de-identification. Do you mean the data labeling, the preparation for the analysis?
So while I think Durga asked that question, I can probably just give a little more context. I think image files could potentially have identifiable information. So that's probably what Durga means by, if the images are supposed to be de-identified. So I can actually quickly answer that. So if it is from TCIA already de-identifies the data, and I think IDC does that as well. And we are actively working on connecting with the imaging data commons as part of the Cancer Research Data Commons ecosystem. So we personally do not have tools to check if it is de-identified or not. But having said that, because the platform is FISMA moderate and we have high security requirements, and of course, we're going to be FedRAMP certified very soon, like I mentioned in the previous talk. Whatever users upload to their own projects are private, so they will not be able to, if they don't want to share it with anyone, it's private to them. And I see Andre has mentioned IDC does not de-identify data. IDC just replicates TCIA data. TCIA has de-identified de-identification algorithms. Thanks, Andre. Yes, user uploaded imaging data. So I think I covered that. And Durga, happy to chat more about it if needed. Okay, the next question we have is, can the ML workflows mentioned here accept CT scans, MRI images as inputs?
Yes, I can answer that. So we are the main deep learning tools support DICOM images and SVS. So we some of the extensions, let's say like that. We support DCM, DICOM, SVS for histology, TIFF images as well. But they are converted in the workflow itself automatically if those are provided on input. And also we have a conversion tool that is also published. It's called the SBG Medical Image Convert. Because these medical image formats are not previewable on the platform, but you can use them to convert them to PNG and then use them like that. But the deep learning tools also automatically support these formats.
Great, thanks, Anna. Okay, so the next question we had is like, is DICOM whole scale microscopy format, WSI, supported?
Yeah.
Okay, yeah. And
I answered.
Exactly. All right. And I think Soner also posted a link to the workflow, which also has that information.
Yeah.
Okay. The next question was, is it possible to configure the workflow to fetch input data automatically using a manifest that refers to the individual items using CRDC UUIDs? So thanks for that question. And I think I tried to answer it in the chat. As of now, we do not, especially for import of image files, we do not have manifest-based import using the CRDC UUIDs. But I think with our upcoming IDC integration, we are going towards that direction. So happy to chat with you, Andre, more about this. And we have prior experience setting this up, manifest-based imports for both our proteomic data commons and the K9 data commons integrations. So we are familiar with how to set it up. So it should be possible if that's one of the user needs. Okay, next question is, are the image processing tool, oh, sorry, I missed one slide. Do you plan to develop workflows that would consume data directly from the CRDC imaging data commons? So yes, that is our intention. Anna, you have anything to add?
No.
Okay, I thought you were going to say so. Okay. We are open to, like Anna just mentioned, we're open to adding any kind of new workflows that will support our users' needs. So, and as part of the IDC integration, adding new tools and workflows to consume data directly from IDC is definitely in the plans. All right, moving on, are the image processing tools open source?
Yes, they are. So,
in terms of, actually the workflows are public, but the source code is not public now. Correct, Anna? I think we just added the work.
Yeah, source code is also available in the tools themselves. You can see that. So SBG tools, you can see that they are under I'm not sure how to pronounce this in English, sorry, but you can see the license under which any of the SBG tools is published on the tool itself when you copy it on your project. So you can see the code, then you can modify it and play with it. So you will find the license and you can find what that means, let's say.
Okay. The next question, I think, yeah, that's just a follow-up. Where is the source code for the tool you just pasted, Sonar? And also, how do I get to the source code of the tools?
Yeah, let me clarify that. Actually, to access the source code, first of all, they should log into CGC and copy these tools or workflows to your projects, then click the edit over there. Then they will see all the scripts behind that. First, they should log into their account, and from the public apps gallery, they just will copy any projects they want, and they will access the code.
Great. Thanks, Sonar. All right. So that was great. Thank you so much again, Sonar and Anna, for the wonderful presentation on the imaging and machine learning topic. So we are exactly at time, which is great. So we can move on to the next topic, which is using the power of cloud computing to scale single cell analysis with the CGC. And we have Dr. Manisha Ray and Dalibor Velkovic talking about this. Over to you, Manisha.
Hopefully you can see my screen.
Yes. You want to go on present then?
Excellent. So hi, thanks for joining me today. And thanks for sticking around for the presentation that we're going to give on single cell research in the CGC. I'm Manisha Ray. I am one of the co-managers of the Cancer Genomics Cloud, and I'll be giving an overview to why we do single cell on the CGC at all. And then I will pass it over to my colleague Dalibor, who developed some of the tools, and he will take you through a live demo of them. So to go over what we're going to cover, we're going to go through kind of the higher level concepts in single cell analysis and in how cloud computing benefits single cell analysis. Then we'll go through the specific set of tools that we have available for doing single cell research on the CGC, a live demo of those tools, and then hopefully some robust Q&A. So why are we so excited about doing single cell analysis? The reason really is that over the past decade, it's become really clear that tissues are extremely heterogeneous. We knew that before the past decade. But the ability to understand that heterogeneity can really lend itself to better understanding the properties that emerge because of that heterogeneity. So things like cancer progression, where the initial set of mutations that cause a cell to become cancerous can be uncovered if you can look at things at the single cell level that would just be masked by all of the other cells if you were looking at a bulk population. Similarly, discovering new cell types, that's very important in developmental biology and the study of cancer progression. And with all of these situations, the very exciting thing is that then you can identify the marker genes for these new cell types and have new targets for interventions for these diseases. So I just have some examples on this slide here of some single cell analysis of the bone marrow microenvironments. So this was done in mice of one of the leukemia types. And you can see in the T-SNE plot here that there's a whole bunch of neutrophils, which one would expect, but the populations of the more progenitor type of cells, you can see a few here, and these red ones here, are much, much smaller. And you can imagine if you looked at this same population of cells in a bulk analysis, these would be completely masked. You wouldn't be able to see them at all. And then on the right, there's something called velocity analysis, which is sort of looking at what cells are going to differentiate into, trying to make those predictions. And you can see that a lot of those progenitor cells, which have these really long arrows indicating their high transcription rate and in the process of changing state, are sort of associated with the progenitor cells, and that they're about to kind of differentiate into the two major cell types present here. So these are the types of insights that you can get to with single cell analysis that were not possible any other way, and can bring about some remarkable insights into many different areas of biology. And this field has only been growing as the technology has made it easier and easier to do single cell analysis. And there's a lot of interest now in doing whole atlases, so whole tissues, whole organisms, et cetera. And there's a wide range of published cell atlases now, from basically all the model organisms, but from very simple organisms like C. elegans, which is pictured on the right here, all the way through individual organs of mammalians, such as mouse. And then there are a bunch of projects in process through the Human Cell Atlas and the Human Tumor Atlas Network that are aiming to get these types of atlases put together for human cells. And in the Cell Atlas, it's intended to be mainly for normal healthy tissue as sort of a baseline comparison, whereas the Human Tumor Atlas Network is looking more at disease states of tissues. And the more atlases more atlases are coming out all the time. The Human Cell Atlas has over 400 publications now. And just another example, the 10X system has over 2,000 populations now and publications. Considering that 10X technology came out in approximately 2015, that's a pretty impressive number of publications. But with these super cool large data sets, they're really large, and data sets like this that are extremely large bring their own challenges. And one of the most obvious ones, which is sort of where we come in, is that large data sets require large amounts of computation. The bigger your data set, the more room you need to just physically download it, and the more computational power you would need to analyze it. And this became very clear back about a decade ago with the advent of the Cancer Genome Atlas project, where this project resulted in an incredibly rich data set where they looked at 33 different tumor types, 11,000 samples, and multiple data types. And it has provided numerous insights into cancer biology. However, it's huge. It's over 2.5 petabytes of data. And this basically means it's not something you can download onto your laptop and analyze there. you need significant amounts of storage to download the data, and then significant computational infrastructure and expertise to actually run anything on data of this size. So that really motivated the transition to the cloud, where these types of, you can avoid download, and these types of analyses become much easier with the distributed nature of cloud computing. So as mentioned by my previous presenters, the Cancer Genomics Cloud allows you to do that by combining the access to the data with the ability to utilize the power and scale and speed of cloud computing. So the platform provides a very stable and secure location to combine basically three things: a way to access data, either these large public data sets or uploading your own data, a place to find the tools. And again, you can either build your own tools or use one of our 600 pre-developed and pre-optimized tools, and access to cloud computing. So taking advantage of the scale, you can scale up to huge numbers by utilizing cloud computing, and it can happen much, much faster than in the traditional computing methods. So the combination of these two things can be very impactful specifically for single cell data sets. So I went through how challenging how challenging the TCGA data set has been to download and computationally access without sort of significant infrastructure. And that is all bulk tissue analysis. When you start looking at things at the single cell level, every individual cell is an entire transcriptome or entire epigenome, an entire genome. And basically every cell can be the size of a single sample in the TCGA data set. And you can see how that would scale dramatically in terms of the size of the data sets you're dealing with. Initially, when the single cell technologies first started being developed, doing one cell was a big deal. But now routinely, people are doing 10 to hundreds of thousand cells per experiment. Like the average experiment has 10 to 100,000 cells. And 10 to 100,000 genomes or transcriptomes per experiment is a pretty significant number to have to deal with. So these big data challenges are just really amplified when you get to the single cell level. So we've talked about why this is so important and how it's difficult, but specifically the single cell data sets are large, they're complex because you're looking at so many different features per data set. And the other real challenge is that these data sets tend to be sparse. An individual cell is not expressing all of the genes that it may usually express at the particular time that you capture and lyse that cell. So there's a lot of zeros in the data, and you have to collect a whole bunch of them to make sense of it. So the data sets are physically large in terms of bytes, and the traditional methods for doing RNA-seq data analysis may not be as functional on these very sparse data sets. To address that, there's been an explosion in the number of tools and also the number of technologies that require different tools for looking at single cell, in this case, transcriptomics. And here you can see a plot from the single cell RNA tools website showing this dramatic rise in the number of single cell tools over the past few years. With this explosion in tools, it can be very challenging to come up with a consensus. If you're not already an expert, how do you know which tools to choose to do your analysis? And that's really where the CGC can help. So with the large complex data sets, the access to cloud computing and the access to the data are both made much easier. Anyone with a web connection can get to these big data sets and analyze them from your laptop without needing any additional infrastructure. The challenges of the sparse data sets can be addressed by the number of tools that we have on the CGC that are specific to working with this type of data, which I'll show in a moment. And the sort of, how do I know which tool of the many that are available to choose? We have a series of tools that are already made, already optimized, so the user doesn't have to do this themselves. And these are all publicly accessible through the public apps gallery. Every app that we have contains a description and benchmarking information. So that can tell you, specifically what biological question these are good for, what, types of data it's good for, and so on. And in addition to that, we also have example data from the Human Cell Atlas and example notebooks that demonstrate exactly how to use these tools. So the CGC provides a lot of tools and resources to make doing this type of very complex data analysis really accessible to any researcher. So now I'll spend a couple minutes talking about the actual tools that we have on the CGC for doing single cell analysis. We'll go through the tools and workflows and then shift over to the demo portion where we'll demonstrate some of the specific analysis you can do with the gene to cell counts. So once you get to that step of the analysis, and then we'll end on an interesting new case study using different types of trajectory inference. And these tools are not yet published, but they're cool stuff we have in progress. So to understand the types of tools that we have available, it's important to understand that there's two main ways that single cell RNA-seq data is produced. The first is through producing full-length transcripts, and that you get the entire, everything that's in that transcript. And this tends to not be able to be multiplexed as well. So the total cell numbers tend to be lower. That typically means that the sequencing depth is higher. With this higher sequencing depth and having the entire transcriptome, you can ask questions that look at mutations, splice variants, and other questions where you really want to see the entire sequence of the transcript. And a very common example of that is doing B and C cell receptor profiling, B and T cell receptor profiling. So looking at the sequence of the variable region of these B and T cell receptors is a very, very popular method. The other type of protocol used to generate single cell RNA-seq data is TAG or UMI-based method. UMI stands for unique molecular identifiers, and that this technology specifically captures the end of the transcript. So you don't get the whole transcript, you only get usually the 3' end. This uses two different types of barcodes, one for the cell, one for the UMI. And the combination of these two lets you multiplex very, very highly. You can do millions of cells in the same sequencing lane. That does mean the depth tends to be lower. So the questions that this technology is better for answering are looking at overall heterogeneity and looking for cell types, not the things you would need high sequencing depth for. To briefly go through a little bit more detail about how the UMI-based methods work. Again, you're only looking at the very end of the transcript. There's a barcode applied to the entire cell. So every transcript from a cell will have the same barcode. So you know which transcripts belong to which cell, and you can multiplex a bunch of cells together. And then there's also a series of random barcodes within each cell barcode that ensure that every transcript has a unique molecular identifier. And what that means is you can control for things like PCR amplification bias and other issues that kind of come up through the library prep and sequencing process. And when you have so few transcripts per cell, this can be really, really important. So generally, you're doing the same steps for both protocols. You're sequencing and then demultiplexing. With the UMI-based methods, you also have a deduplication step, then align, and then quantify. And Dalibor will go into those steps in a bit more detail. So for alignment, we have a series of tools, including the Cell Ranger suite of tools. So if you have access to the license for the 10x tools, you can use them on the CGC. We also have some tools based on pseudo alignment, which we found is better for certain situations. And as I mentioned, there are a whole bunch of different ways to generate the actual single cell data. And with the combination of tools available, we can basically support all of the ways that you may prepare your single cell RNA-seq data sets. So a bit of terminology here, which has been mentioned by several of my other colleagues, is that when we talk about data analysis on the 7 Bridges platforms, we generally use the terms tool and workflow. So tool refers to a single step of an analysis method. So something like demultiplexing could be a single step. A workflow is a series of tools that are all strung together into a pipeline, which could be something like taking in the raw data and exporting the number of genes per cell. And we use those terms a lot when we talk this through. The third type of analysis we have, which has been demonstrated again by some of the previous presentations, is interactive analysis. So this allows you to use your own Python or R notebooks to run interactive analyses in the cloud in your same project with your same data without needing to download it, and do it interactively. So this is a nice way to get the visualizations that you will often be looking for with these types of analyses. So here's sort of the full list of what we have available. We have, again, several tools for working with full-length data. SmartSeq 2 is kind of the standard one people use for that. And then we have methods to do the B and T cell receptor profiling. We have a series of tools for doing alignment. And again, that can be done with the Cell Ranger 10x toolkit or with some of these open source methods. And then we have a series of workflows for doing tools and workflows for doing tertiary analysis, which we will be talking about more in demoing on the next section. And then we also have a series of notebooks for doing these interactive type analysis. So we have an interactive way to do batch correction, an interactive version of Seurat, which is a very popular tool for clustering the cells and detecting the important marker genes. And then, as I mentioned, in development, we have some tools for doing lineage tracing type analysis, which we'll be going through at the very end. And all of these tools are available in our public apps gallery, where we have over 600 tools that are pre-optimized to work in the cloud. And you can filter through them with a bunch of different categories. We have a single cell category, and then you can easily see all of the tools that we have readily available to be used for single cell analysis. And with that, I will pass it over to Dalibor to talk through actual use of these tools.
Thanks, Manisha. I will share my screen now. Is my screen visible?
Yes, I can see your screen.
I'll start presenting. So hello, everybody. My name is Dalibor Velikovic, and I will be talking about downstream analysis that we perform on gene-to-cell count matrix. So basically, this matrix is the output of a quantification tools, alignment and quantification tools such as Cell Ranger. And it's basically a table that holds information of number of reads assigned to each gene per cell. This matrix is a starting point for some of the tools I will be talking about, such as clustering and detection of marker genes, pseudobulk differential expression analysis, and trajectory inference. First two of them being our public pipelines, while the third one is interactive analysis, still under construction. Our tools are built by using several single-cell ecosystems, such as Seurat, single-cell experiment bioconductor, and Python's ScanPy. And before I talk more about these tools, I will briefly go through the usual flow of the single-cell analysis. So this flow can be roughly split into the pre-processing that includes quality control, normalization, highly variable gene selection, and batch effect correction, basic data analysis with dimensionality reduction and clustering, and also higher level analysis such as pseudobulk differential expression, marker gene analysis, gene set enrichment analysis, velocity, and trajectory analysis. All those steps are better summarized in this diagram, and we will start from pre-processing. So the starting point is our gene cell count matrix. So in this matrix, the columns are cells, the rows are genes, and for example, 14 means that there are 14 reads aligned to gene 3 in cell 2. The first pre-processing step is quality control, where we aim to remove the low quality cells. And we do this based on several criteria. We will remove each cell that has a low total count, that has a low number of genes with non-zero counts, And also we remove the cells with the high count for spike genes and mitochondrial genes, because that means that cell membrane was destroyed. The next step is normalization, where we aim to remove technical variation induced by PCR amplification and reverse transcription. And we do this by generating cell-specific scaling factors, which we use to scale our counts for each cell. After we have done those two steps, we have this matrix only with a fewer cells, and the counts are now normalized. Still, our matrix has a lot of genes, 30 to 40K usually, and because in most single cell data sets, almost all variation is contained within a few thousand genes, we use mean variance trends to detect only highly variable genes and use them in the further analysis. Finally, if our data is coming from multiple sources, such as multiple labs or protocols, data probably contains some kind of technical variation induced by different library prep, and we can use batch effect correction tools to remove this technical variation. Coming next, we start our analysis with a dimensionality reduction step, which starts with principal component analysis, which is basically spinning our coordinate system to fit as much variation as possible into the axis that we call top principal components. And we use those to further embed data by using UMAP or TSNI into 2 dimensions, and we can use this for data visual inspection. Next, we use PCA components to do a population analysis of our data, and we do this with the graph-based clustering methods, where the first step is generating our graph, and for single cell, it is a shared nearest neighbors graph, where cells A and cells B are connected only if cells that are close to cell A in PCA space are also close to cell B in PCA space. After we generate our graph, we take advantage of this graph structure to detect those nice interconnected components that are called communities, which usually present cell types or cell subtypes. And we can do this with any community detection algorithm, most popular being Louvain. Once we get here, we can go into one or multiple directions for further analysis. The most popular one for sure being marker gene analysis, which present differential expression testing between cells belonging to different clusters, and which results in genes that are characteristic for each clusters or that make difference between chosen clusters. Also, if our dataset contains multiple samples and defined conditions, we can do pseudo-bulk differential expression analysis to infer what's different in expression between those given conditions. And if we know that our dataset contains some kind of cell development or differentiation process, we can take the advantage of our graph structure to perform trajectory analysis with tools such as Slingshot. For example, if the yellow dots were our starting populations, based on the graph structure, we could infer that, for example, it splits into blue dots and orange dots, and the differentiation continues to green dots and dark blue dots and so on. And if we also have available spliced and unspliced counts, we can take advantage of the fact that all the unspliced reads will become spliced at some moment. And by using this fact for every gene, we can derive vectors for each cell that show the probable direction of cell development. Finally, for all the methods that yield some kind of differentially expressed genes, we can use gene set enrichment analysis with publicly available data sets of curated gene sets that are related to specific biological processes to make more sense of our data. This wraps up the basic processing. Now I will be talking about our first pipeline, clustering and detection of marker genes with Seurat. So this is a single sample pipeline. So the pre-processing is pretty standard as explained, only without batch effect correction because we use only one sample. Basic analysis is also standard, and the pipeline ends up with the marker gene analysis. on these pictures, you can see the example of the Seurat output. So this is the UMAP embedding with Seurat-derived clusters. And on the right, you can see the same clusters, so the same cells, only annotated with a single R package. And you can see just how well those clusters correspond to the actual cell types. So the biological question we solve with Seurat is what cell populations are present in our data set, and also what genes are characteristic for each of our clusters. Now we will do a quick demo with Seurat. So it can be found in our public apps gallery, and you can find it by searching. So here it is. We can copy it to our project and find it in apps there. We choose our app and click run. So the main input is the compressed outputs of the Cell Ranger tool, which we select. We tell algorithm that the count matrix was generated by Cell Ranger. After that, we have available some pre-filtering steps, which will leave the default values. We also have some QA settings, which we will also leave at default. Next, we set a very important parameter, which is the resolution for the Louvain algorithm, which controls the granularity of the clustering. And another, we set the same resolutions for the class tree algorithms that gives the relation of the clusters generated at different resolutions. Next, we click run. And the task goes to the queued state. And after that goes to the running state. So because task takes some time to finish, we will go to the completed tasks project and find the completed task there. And here you can see the important outputs. So the first one being the plots of marker genes for each cluster at each resolution, differential expressed genes for clusters at each resolution in tabular format, RDS object that we can use in our further analysis, and also a brief report, which we will go through. The report starts with a brief introduction to the steps in our analysis. After that, we have a section on quality control and removed and filtered cells. Next, we have plots of cell cycles to see if some cluster is characterized only by cell cycles. We have the choice of number of PCA components used in analysis. And after that, we have clusterings at different resolution. At lowest resolution, the number of clusters is minimal, 10 in this case. And as resolution goes up, the clusters split, for example, cluster 3 splits in 5 and 3. And finally, we have class tree that, for example, gives us just that information that cluster 3 goes into 3 and 5. And finally, we have the tables of differentially expressed genes. And also this demo was performed on a 10x squamous non-small cell lung carcinoma tumor dataset. The dataset contains around 7,000 cells and was processed in 24 minutes for a fraction of $1. The next pipeline we will be talking about is pseudo-bulk differential expression analysis. This is a different pipeline. The biological question it tries to answer is if we have, for example, 6 samples, 3 tumor samples and 3 normal samples, what's different between those conditions. So it's very similar to the bulk RNA analysis. So the actual differential expression testing is performed on pseudo-bulk expression profiles that are generated by summing counts for cells together that belong to the same sample. And why are we using pseudo-bulk? Well, we are using it to reflect the fact that biological replication happens at the sample level here, and this also helps with modeling simplifications by masking variants that would otherwise exist within those pseudo-bulk samples. In the end, we do differential expression testing by using EdgeR package. So here is the full pipeline. The pipeline starts with the basic processing with batch effect correction for multiple samples coming from different sources. After that, we perform basic data analysis, and the pipeline ends with two types of pseudo-bulk differential expression analysis. First one being pseudo-bulk analysis by using all the cells. And the next, we also do pseudo-bulk differential expression analysis in cells belonging for each cluster. So for example, imagine that out of our 10,000 cells, 2,000 cells cluster together and represent some endothelial cell type, for example, and those cells contain cells from all the samples. And by doing differential expression testing only in those cells, we can see what's different between tumor and normal in cells of a certain type. Next, we will do demo on data set that contains 4 samples for tumor and normal in BRCA1 carriers. This app can be also found in our public app galleries, and we can copy it to our project where we can run this pipeline. The first input represent the multiple outputs of a cell ranger or some other aligner, one output per sample. We also need a metadata table that connects our samples to batches and conditions. We set input type to be cell ranger. We leave the pre-filtering parameters at default values. We choose our integration method. So 2 integration methods are available, Scanorama and BBKNN. Scanorama usually yields better integration results, but in this case, we use BBKNN because it's better at handling the large data set, such as this one, which contains 80,000 cells. Next, we choose that our species is human. We set clustering resolutions for Louvain algorithm. We choose to generate a class tree and also to do differential expression testing in clusters as well. We click run, and while the task is starting, we can also take a look at metadata table. So for each sample, we have sample name, we have batch information for batch effect correction, and also information on condition for testing. Because the task takes a while to finish, we will go and preview the results of already completed tasks. So we have differential expression results for testing inside clusters for each resolution. We have differential expression results for testing in all the cells. We have HTML report, interactive report for better data exploration, and also single cell experiment object for further data analysis. We will briefly go through the report. It starts the same way as URAT with the explanation of steps. We have data on cell pre-filtering for every sample, quality control plots for every sample again, mean variance trends for each sample, batch effect corrected data in the UMAP plot with batch identifiers, class tree that gives us relation between our clusters at different resolutions, and clusters obtained at each resolution. Finally, we have table of differentially expressed genes between conditions when testing on all cells, and also tables of differentially expressed conditions genes when testing on clusters at each resolution. So we can go back to the pipeline. So this pipeline contains 80,000 cells and was processed at 49 minutes for a fraction of $1 again. Finally, we will show example of trajectory analysis at our interactive analysis. So we did the interactive analysis on data set where researchers injected the melanoma tumor cells into mice, and they followed the changes in the tumor cells as well as in the healthy cells. After 5, 8, and 11 days, researchers focused on T cells and fibroblasts associated with tumor. And in our analysis, we did the analysis of endothelial cells. This data set was produced with SmartSeq2 protocol, and we accessed it through Cancer Genomics Cloud platform. And data set consists of some 12K cell files. To align and quantify this data set with HiSET2 and RSAM, we used SmartSeq pipeline that is available on our platform. And we aligned and quantified 1,000 cells, which was done in 49 minutes on 10 instances for $5.5. For trajectory analysis, we used slingshots. Slingshots require standard pre-processing of the data. After that, Slingshot takes advantage of our clusters and their connectivity to form a cluster connectivity graph. Example of this graph is here, and it fits the minimum spanning tree to this graph, meaning the set of edges with maximum weights that go through all the clusters but make no loops. And if we know our starting cell populations, we can infer, for example, that F is first branching point and data goes to A, C another branching point, data going to D, and after that B and A. A problem with the trajectory analysis often is not knowing which is the starting point. So when we processed our data set, we did basic pre-processing with Surat, but we used velocity analysis to find which cluster is a root cluster, and we identified cluster 2 being a root cluster because all the cells try to escape from this cluster based on those velocity vectors. After that, we used the slingshot with this information available to generate trajectory through our data. In the end, we also used the TradeSeq to identify genes, and we displayed some of them which are involved in pathways related to DNA damage. This wraps up our presentation. Now we will make a short summary. So we covered general flow, single cell analysis, and also specific analysis such as marker gene analysis, pseudobulk differential expression, and trajectory analysis. There are multiple ways those can be used in tumor research. For example, you can research cell populations in tumor and its microenvironment, and also characteristical genes. You can do differential expression analysis between various conditions, for example, samples treated with various drugs, tumors and normals, different clones, and so on. And you can also track progression of tumor cells with analysis such as trajectory and velocity. And of course, you can analyze your data on our platform. You only need to upload data and use our aligners to align your data, and tertiary analysis up for further analysis. Thank you for your attention. If you have any questions, feel free to ask.
Awesome. Thank you so much, Manisha and Dalibor, for the wonderful presentation on single cell data analysis. We have one question from one of our attendees, and I should first thank all the attendees who are still there with us, and and then for posting your questions here. So the batch effect correction, is that not equivalent to integrated analysis in Surat? For example, find integration anchors or integrate data functions.
Yes, Surat can also be used to integrate data, but in our pipeline, we chose to use methods such as Scanorama or BBKNN based on the benchmark papers for single cell batch effect correction tools.
Okay. Thanks. So while we're waiting for others to post their questions, I have one more question. So are there any standard pre-processing criteria that should be used for single cell processing?
Pre-processing criteria, so we can go to the slide. So the mandatory steps for sure are quality control, because you have to remove bad cells. and normalization, because there's always some artifacts connected to PCR amplification and reverse transcription. And highly variable gene selection solves two problems. Noisy genes, which the majority of genes will only carry technical noise. And this will also take care of sparsity in data, because top genes will be less sparse and therefore easier to analyze. And of course, if data is coming from different labs or different protocols, for joint analysis, batch effect correction is also mandatory. But be careful, you can only do this if there are some shared cell populations between your data set. If you have one batch of data A, which is totally unrelated to one batch of data B, you will not be able to integrate the data with no method.
Got it. Actually, that was one of my other questions, like when should we use batch correction for single cell and when we should avoid, and you've already covered that. Okay, we have one more question from our attendees. Are the cells spatially localized? That is, do you know which ones are close to each other in the original tissue?
No, this is not a spatial analysis. The cells here are grouped only on based on the similarity of their expression profiles.
So I can expand on that a little bit more. So everything that you're seeing here is not spatially located. Generally, all of the single cell processing techniques require you to make a single cell suspension, and then you lose all localization. But there are all these really cool technologies coming along that enable you to do some level of transcriptomics or gene expression in situ. So like highly multiplexed fish, for example. And there are tools to work with that as well. So that's something that also could be handled by the CGC. And it's something that the research is generally going to. Often what people do now is they will, like, in parallel have a tissue slice or, you know, the full structure of the cells that they can stain or otherwise look at to understand that spatial context. So right now, those questions are answered in two different experimental methods, but in the future, it should be possible to do it together, and it should be possible to analyze it on a platform like the CGC.
Great, thanks, Manisha. Okay, so this was wonderful. Thank you, everybody, again for attending all the 3 sessions, and we're very excited that our very first summer symposium started off very well. So if you have any questions, you can always reach us at support@sbgenomics.com. And also you can go to our website. There's a help page which gives you the email address you can reach if you have questions, and also a link for office hours, which happen on Tuesdays and Thursdays. And lastly, the slides and the recordings will be posted in the website, and you'll get an email when they're available. Next Wednesday, we have our regular CGC monthly webinar. We have an interesting presentation by one of our collaborative project awardees, Ms. Margaret Linian. She is from Arizona State University, collaborates with Dr. Valentin Dino. So you're going to hear about how she has used the Cancer Genomics Cloud for pan-cancer translational bioinformatics research, and how she has used the platform for detecting and ranking mRNA dysregulated pathways. So I hope you will join us next week. And I would like to thank all the speakers for all their hard work and for giving excellent presentations and live demos. Bye, everybody.